We are going to our next session, that is preclinical studies uh, with animal models and anti-cancer agents. So I think, Pooja, we can start this one. And many thanks for your, uh, that is in the midnight in USA, and you are with us and giving a lot information, and that is all the expert lectures, madam. Many thanks to you. So Pooja, please start.
Thank you, ma'am. Thank you, sir. We can start now our lecture 8 for GIAN 2025 on preclinical studies with animal models and anti-cancer agents by Professor Niharika Nath, madam. Ma'am, you can start now.
Thank you, Pooja. So this next topic is preclinical studies and preclinical studies with animal models and with an anti-cancer agent. And so um, animal models are always just preclinical. So I need not have stated that it's a preclinical studies, but just for you to understand that it means preclinical. So things that are still in research, things that are still in the lab, is preclinical. Everything that is before clinical is preclinical. And so really it's really about some animal models in tumorigenesis. And to study if any new treatment that you have thought of by research, any new modality of or different drug design, derivative, anything that would be required needs to be tested out in an animal. And so the requirement of having an animal model is just that there is some kind of in vivo exposure, in vivo experimentation, in vivo assessment. In vivo means um, in an intact organism. And so the closest we are able to do on a regular um, basis with some degree of comfort is with rats and mice. And so today I will talk about these just a couple of animal models, the most important ones for cancer at least. And maybe just take one example. Either I can talk about, so I will talk back and forth about colorectal carcinoma or hepatocellular carcinoma as I'm showing you. Although I'm not showing any colorectal, but I might refer to that as well. So this diagram is of a liver, and it has been resected. This liver has been resected from the animal, from the mice, and it shows a tumor. Okay. So, oops. So I took out some sections from here. There was a paper that I had put in, but I thought it was a bit too much. I took that out. So And so let's just take a look at these, the overall thing. So first of all, in order to have animal models, first of all, you need to understand exactly what kind of a system you're dealing with for this cancer. For colorectal cancer, you should know about all the locations and understand well the physiology of the GI. And for liver cancer, it is important to know the bulk or the type of the cells that are in these. So this is just a structure of the liver, and the liver has a hepatic artery and a portal vein, and as you can see here, there's not much for us to really discuss. This is more of a physiology, but is made of hepatocytes. There you go. So it's made of uh, hepatocytes. Sorry. So it's made of. Are you able to see? I don't know what happened here.
Yes, yes, madam.
Okay. So it's made of hepatocytes. So it's kind of important to know what kind of the cells are in there. So that was just really my point here. And so an understanding of the nature of the cells and what kind of cells it has. So for colorectal cancer, you would need to understand what kind of cells that you are dealing with. So these are cells of the colon or the cells of the rectum, and so there are several ways of going about this. So anyway, back to the liver one. All right, so because we would like to be able to study everybody all over the world is just trying to study novel therapies. And so when they're looking at this, they're looking at how the cancer spreads and what it is that the cancer looks like in the liver, and so what it is that can be done. So when this is examined, then what is done is typically some cells from the diseased liver are taken out and put into cell culture. So here you have a hepatic carcinoma that you can see this, and it's probably because of some Hep B or Hep C viruses, or maybe it came because of alcohol cirrhosis, etc., and led to this kind of a carcinoma. So, whatever it is, some cells have to be taken out and they have to be then completely put into tissue culture. So, even though I'm not talking about it's only about animal models today, the bottom line is that you still need these cells in tissue culture. So, a human who has the cancer, that human who is the patient, their cancer cells, that cancer cells are taken and then they are grown in the laboratory under cell culture. And even for studying for the sake of animal model, using for animal model, that's how it is done. So, so I will just ignore this particular slide about what were how we did that, how we got it. So we can get it by biopsy even if we don't do a full surgery and for the cells, but we don't need to go through these treatment options. It's just that you need to understand that if it's in the liver, then there's not there's not much, a lot of surgery needs to be done, a whole new liver needs to be done, etc. It's just to give you the sense of the whole story. And liver cancer, once the cancer really enters the liver, I chose this one only because it is, there's not much that can be done. So, and typically, when the cancer spreads from the cancer has spread here, to the liver, that's kind of like almost the end game. There's not much to do. So once if there's any metastasis here, but what we are talking about is the primary cancer, the Metastasis is not what we're talking about. So talking about just the primary cancer. So let's go to after we take those cells from a human being, there are uh, a few options. Number one is you continue to do research in the tissue culture model. So a tissue culture model, which I have not shown you here, is the norm. Why? Because in a tissue culture model, you can see here, you see these cells that they're in tissue culture here. You're not hurting anybody. You can continue treatments, examination treatments with new drugs, and you can do whatever you want to check. And so um, you're not taking in too much resources. So let's say that you did find a potential a drug. There is a potential drug, and that potential drug is showing promise in tissue culture. So if it has shown promise in tissue culture, then then if it has shown promise in tissue culture, then what should happen is that it should be examined next in these mouse models. Now, in a mouse model, why? Because in a tissue culture, it is just one type of cells. In tissue culture, you just have one type of cells. There is nothing more. It's just these liver cancer cells, and that's all you're checking. So, in order to put it into the animals, we need to now really think. We need to think how much, because animals are also expensive. Also, there's a cost effectiveness, and it's a life that you are sacrificing for the sake of the study. So, uh, so uh, in mice models, there are various ways to study mice models, and the the first thing is that in a mice model, you need to, if you would like to check the effect of the drug, then you need a mouse that will have the cancer. So for the first model, which is less expensive and not bad, because it not bad at all and doable as a first time experiment, is called as a chemically induced mice model. In a chemically induced mice model, the mice will never get the cancer, but they are given a special chemical agent to get exactly the cancer that you are looking for. So, a chemical, a typically they would be like a environmental carcinogen. A environmental carcinogen that we have studied, that people studied beforehand, and examined it and injected into mice. So you see that in a chemical model, it has to be injected in, and it is just injected in, and you have to give the mice some time, a month or more, to start showing the signs of the cancer. So in this mice model, a chemically induced mice model is is that mice model in which you get the closest effect simulating the cancer that you're looking for. So, not all chemically induced models are good, but if you had nothing more to test, if you there was no other way to get a cancer in the mice that is similar to the human uh, liver cancer, then what to do? So the only thing to do is to people have spent years on this, is to take a environmental carcinogen or a compound that people figured out will cause the cancer. And so people tested 10 different compounds on 10 different mice, and then after a month or so, or did it in rat. And after, rat, it would take, let's say, a month or so. Mice, it would take, let's say, 2 weeks or so. And and when they did that, they waited, and then they um, sacrificed all these mice, and then they looked open inside, and they saw which one has the liver cancer that is very similar to human. Uh, does the liver cancer develop? Then the mice model, the question is that in the chemically induced mice model, did the cancer develop in the brain also? If it did, then that's not a good mice liver cancer model. Then they would look at the organism. Did it have any other effects? Did it have, did it damage any organ completely? No cancer, but it damaged, maybe it damaged the kidneys. Then that's not a good mice model. So after all of these years of study, people found, and this is not recently, way back in the 70s, that there are one or two or just three chemical agent that exactly gives us the kind of cancer that would happen in a human being. And so that's chemically induced with DEN, produces hepatocellular carcinoma just like it would in human, and it does not produce a cancer of the brain. It does not produce in the same time when that cancer is there. So we don't want a complicated system. So there is no cancer anywhere else, and it is only cancer in the liver. That is the mouse model that can then be chosen. So, and when you have this mice model, then you tell the whole world that I have a mouse model, and then everybody will start doing what you did, and everybody will start, some company will start making it, and then people will start buying it, and then they will tell you, okay, you got the mice, now you inject it with this carcinogen, and in, let's say, 1.5 to 2 months, or 1 month, you will later on, you will get this particular, the mouse will get the cancer. And now you can feed the mice whatever you want to. So, for example, let me take a slightly side discussion. Let's say there is our favorite, our favorite dietary agent, our favorite dietary agent, that is turmeric, hulud. So turmeric is known to be anti-cancerous. Turmeric is known to be anti-cancer in the colorectum. So colorectal prevents colorectal cancers. Long-term use, not one day, not 2 days, not 5 days. Long-term use. So if you wanted to check turmeric now, you would take that mice model, you would inject it, you would wait for whatever the time is, or the rat model, and you would wait for whichever time it's is given for the dose, 3 weeks or a month, and then the tumors would develop in the liver. And all this time you can keep on feeding it because it's a dietary agent. You keep on feeding it the potential anti-cancer agent. So you keep on feeding it that potential anti-cancer agent. And then 4 months later, you have control mice. You have control mice who have tumors, and you have your test mice who have tumors and have turmeric in their diet every day. And then after 2 months or 3 months, you sacrifice the mice and you see what happens to their tumors. So that is your chemically induced mice model. We're not lucky to have chemically induced mice models for everything, but for colorectal, for HCC, hepatocellular carcinoma, liver cancer, yes, we do. So therefore, that it applies. Now, there is a, people have a problem with this. Why would you have a chemically induced mice model? Because in humans, maybe it might be different, No, in humans, maybe it's not induced from a carcinogen because I had a mutation from something else. Anyway, the bottom line is a chemically induced model is has its own problems only because that chemical agent creates other cancers as well, and then there's this complication, and you don't know, the complications become too much for observation. So next up would be the xenograft transplantation model. A xenograft is a better way to study it because we want to study human cells. So far in chemical induced model, That was completely mice. And there are some differences in mice and humans in certain cells. So in a xenograft model, okay, as you see in the right here, what is being done is that you take those cancer cells from the human being that who has cancer. You take it from the human being who has cancer and you grow it in cell culture. So you take it from the patient. And so we don't have to take it from the patient every time because these cells from the patient, they're already, they're already growing in the lab. The whole world has it. We don't need to go to the patient right now, but we already have these. All right. So, unfortunately, this is not the mm, liver here in the picture. So it's the lung. So we can take these hepatocarcinoma cells, and we have to grow them in cell culture, and then after we grow them in cell culture, we inject them into the mice underneath the skin. Okay, so we can inject them underneath the skin, or we can inject them straight into the liver. So it's giving human cancer cells directly to the mice underneath the skin, near the leg, near the near the leg, or do a little surgery and go straight inject it into the liver. And then in a short amount of time, there will be the cancer. So that's why it says xenograft. It's a graft from a human and transplanted, sorry, into the mice. So it is a xenograft, and obviously you know that there's going to be a tumor that's going to be formed. And so once that tumor is formed, then you can give the mice your favorite turmeric or garlic or whichever it is through the diet. So diet is the norm here. You feed the mice either a dose of whatever you want, or you just put it in the diet. And so turmeric could be fed in the diet even from day one or later on or after the tumor has been formed. And then about 30 days later, typically the standard is about 30 days later, when the tumors have grown and you have batches, you have some mice with tumors, some mice with the tumor and the anti-cancer agent curcumin. And then you sacrifice the mice and then you look at the size of the tumors. So, and that's how it would be done. And so that is also good, but so why is it good? Because all the tumors are inside the body, everything else is there, all other cells are there, and it's human cells. So it's something a little bit close. And again, these mice, they do not reject those tumors, so they have to be mice that have a immune defect such that they will not reject it, so that you can do the study. And still, it's not the best. So the best one would be, why not have a mice that will automatically get the cancer um, without anything from outside? And so that is genetically engineered mice. And they will have genes. These mice have been made, and the genes are already mutated. So that's how they're made. They already have mutations in the genes that we know already. So you have to know a lot about this cancer and with genes. And then the mice will automatically get its cancer after, let's say, a month, and then you can treat it with whatever you want to treat it with, and then give it another month. And then you will have two batches, one batch in which you have tumors but no treatment, and another batch in which have tumors and treatment. Then you sacrifice the mice and you look at the size of the tumors in the liver. And so pretty much what I said here is all in the same diagram, just all of that is just too much written here. So I'll just move off. Okay. And so in some of these, so this picture here is not for, this picture is not for hepatocellular carcinoma, but I just wanted to show you that you can do a xenograph for prostate cancer also. So in a xenograft model, it can be termed as orthotopic. Orthotopic xenograft model means you take the cells from the human, the from the human prostate cancer, and you don't do it immediately. We already have those prostate cancer cells in our freezer. We always keep them because we're doing research with them all the time. And so it's not like I have to go to the patient today to get it. These cancer cells are immortal anyway. So, so in an orthotopic model, instead of putting it under the skin, we put it straight, inject it into the prostate or to the organ, and then you grow it for, let's say, 2 months or so, because you have to let it grow, and then you treat the mice. You feed the mice with whatever your anti-cancer agent is. Feeding is a very good method. It's called as gavage. You force feed, or you give it in their food, so you can give it um, whatever new agent that you have discovered. And then afterwards, after about 30 days or 60 days or so, 30, 45 days or so, you can remove whatever needs to be removed, and you can do a surgery, and you can look at the organs, etc. So, but the difficulty is that you need to be a good surgeon in order to do kind of like inject it at the right place. Um, so major benefits, so I'm going to come to a couple of examples. So major benefits for using mice model. So one of the major benefits is that, in mice and human, the organs and the locations are very, very similar. So that's why we tend to use a rodent. They are small, and mice have a small life cycle also, short life cycle. So a lot of the genes, sorry, a lot of the genes are very, very similar between mice and humans. So there's a lot of conservation that goes on. And then uh, in terms of mammals, it's one of the best ones to really look at, if considering that it's small, but it's a mammal. And so this is one of the better ones to look for. So mice model. So in order to understand it, you need to really appreciate the similarity of the genes. Okay, so the conservation of the genes. And the very bigger important is that for hepatocellular carcinoma and others, for those that have solid tumors, not for the blood tumors, really for the solid tumors, uh, the all the other tumor environment and all the other supporting stroma cells, everything is pretty much there, and we can stud they will be a part and parcel of the process. And so therefore that's an important thing. And we tend to, when we do tissue culture, we tend to get excited when we see a certain result, but a lot of the time it should always be taken with this concept that tissue culture may not match what happens in vivo. This is in vivo, because the tumor microenvironment is not there. There's no immunology, there's no immune cells. And so a lot of the tumor microenvironment, even the blood vessels, etc., all of that is important. So on the left-hand side, you see there are endothelial cells, there's angiogenesis, there's all these different kinds of secretions that are the liver is going to make to grow. Um, and then there are the all the other chemicals that are trying to hinder, and then there are these surrounding cells that will, including blood vessels, that are part and parcel of the process. And so some of these we are able to test. We are able to test various kinds of other compounds. Okay, so we're able to test the at the preclinical level. We are able to test for right dosage, for right efficiency, and for side effects. So together we use them as a, not really as a, although I said here clinical assessment, but we can use this for preclinical assessment for various kinds of other tumors that are in mouse models. So all you have to do is just take these mice models of various types. They're available in the market. They have been stimulated, and these various mouse models can be tested with other things because our treatment mechanism, remember, we are feeding the mice the drug. So it becomes kind of easier to test a lot of other things that you had, that you wanted to test because you don't have to inject. Once you've injected the cancer cells in, the rest is treatment. So it would be really like a human being would get treatment. So, so the bottom line is that the mice model has to be immune deficient, and immune deficient so as not to reject it. That's the only thing. So it would be immune deficient so as not to reject it straight away. And that is a problem still because we are still using some kind of mice that are not going to exactly represent the natural condition. So that is just something to keep in mind. So the most easiest and the most affordable xenograft mice model is the ortho topic. Sorry. Um, Uh, mm, so these and it is done in a sorry, the subcutaneous mice models, but they are done. Everything is typically done underneath the skin, and it takes the tumor that grows right underneath the under the skin is a tumor that represents the human tumor, and the mice kind of like carries it around, so it carries it on its skin near the hind leg. All right, and it just carries the tumor around for a while. Okay, so that's the first choice, and then comes the orthotopic, which is at the site of origin. So, and that is the second choice. So once you've done the xenograft subcutaneously, then and you've gotten your results after 2 months or so, then you would like to do a orthotopic one because this is the real disease situation. It's just that it needs an expert who knows how to find the organ in this small animal and be able to inject it very appropriately as the case. And so here's a picture of a tumor on the left-hand side of a we don't call this, it's a tumor, we're calling it tumor, but it's a xenograft tumor that is now growing. And this is kind of like small, it can be fairly big. And others are near the shoulder area that you just see, and so they can be on that location. They don't go to that location, they were injected at that location. So the foreign xenografts, the tumor cells, the cancer cells were injected there, and so they grow there. So this is a animal carrying a human tumor. Okay. Now, a syngenic mice model is one that has a very intact immune system, and it does not have a rejection going on. And so this is a different kind of a model, and it can be, because this is a not a mice that has its immune system kind of like destroyed, um, but or inhibited. So this would be the right kind of model to study a cancer immunotherapy. So, and so this would take a longer time because Um, oh no, not this one. This is still coming from the culture. It is not happening on its own. That's for the next one. And the tumor would just the tumor can be injected at at various locations. So it can be done in the tail vein, it can be done under the skin. Skin is the most popular one, and it can be done also in the brain, and only because it has a fully functional immune system. And this is the one that you should study for metastasis. For metastasis, this would make sense because we know that in order for the cells to grow somewhere from one place to another, it is manipulating the immune system. So this would be the perfect good model to study the immune system. And so this is called as a syngenic mice. So in a allo transplantation, if you can see the bottom, I have a spelling mistake there. In a allo transplantation, the transplantation of the tissue is from the same species. So it will be a cancerous tissue from another mice that is injected. And that would be a real way to study it, although human cells would not be involved here. But that would be a more natural condition to study. And so then once this is done, the metastasis occurs. Either you can feed the mice the drug, the compound, or you can provide it in any other way that you choose, but feeding is probably one of the best ways to give to see if the treatment works. And so we've talked about many of the advantages already. Uh, the most important thing here is that the models, this in vivo model, is extremely important because you can get you can have all these similar situations to the mutation, the tumor environment, um, the blood flow, the angiogenesis, etc. All of those would be very much intact. So they are very reflective of a start of the cancer and also for metastasis. So for prostate cancer, this is true also for the other colorectal and HCC. And and so this is just a higher cost. It's just a long time. That is what happens. And xenograft, at the end of the day, if you're transplanting a human cell into an animal, I mean, there are some questions that still can arise there. So now to show you what is a prevention treatment model. In a prevention treatment model, I want you to look at the left top. In a prevention treatment model, um, you want to see, um, you want to do cancer prevention. Now, we all know in today's date that a lot of the cancers are occurring because there is a long time exposure to some environmental carcinogen, or maybe to, we don't know, maybe pesticides in your food for a long time, or maybe radiation, so certain kinds of radiations. So all these kind of, like, we know that there are some cancers that can be preventable. And so if they can be prevented, then by intervention, what I meant by intervention is that taking a small dose of some compound before the cancer even starts. So so before the cancer even starts, maybe if I take, this is the theory, if a human being takes for example, curcumin, no, sorry, not curcumin, but turmeric. Turmeric has the compound called as curcumin. So let's say holud, for example, turmeric um, is can be taken for a lifelong time. And so people ask the question, well, maybe is that the reason why in the colon and the rectal there's not so much of cancer in Southeast Asia or in India at least? Yeah, maybe so. So a preventive compound can be taken. So look at this in an animal model, which is going to run mice xenograft model, which is only going to run for 5 weeks. The first week, you give the drug. So here I have in the first week, I give the drug. Look up here on the left. The drug is fed to the animal from this week, a week even before you give the cells, cancer cells. So that, and so then after 1 week, okay, everything is, we're trying to shorten everything that we had happens in human life into 4 weeks of the mice model. So after 1 week, you can inoculate the cancer cells. Inoculate the cancer cells means inject the cancer cells underneath the skin and let the tumor develop. And every day, just keep on feeding it this drug, giving it this drug that you think can be used for prevention. And so every day you're feeding it the drug and you are checking the size of the tumors for 4 weeks. And then you sacrifice. Whoops. And then you sacrifice the animal, and the tumor is on the skin. It's a big tumor. So you have 4 or 5 mice that have the tumor, no drug treatment, and you have about 5 mice that, only 5 mice. You don't have hundreds of mice here. You just have only 5 mice, and they have also been injected and they're developing the tumor, but they do not get the drug. So two parallel groups, two cohorts. And look at the bottom graph now. In the bottom graph, you see that the red shows you the control, that after 25 days, there's a the tumor size is on the y-axis. The tumor volume is fairly big because this is after surgery. You have to take out the organs. And so the tumor that is on the skin of the animal, that is fairly big. But those that are treated with the drug, so you see here the blue line, the tumors are small. How did we measure the tumors? We cut open the animal, we sacrifice the animal, and take out the tumors, and we look at the tumors and we weigh them. We measure them with the vernier calipers and look for volume, etc. And so that is the way to know that it has really worked. Okay. So, in a treatment protocol, so that was a prevention protocol. In a treatment protocol, you will inoculate the cancer cells first underneath the skin. And then after 7 days, you would give the first dose of the drug. So you would give the first dose of the drug, and then every day you keep on giving it the drug as necessary. And then you will start recording, because you can feel the tumor on the skin on the outside. Remember I showed you. So you keep on measuring the volume with the vernier calipers, and then at 25 days or so, you sacrifice, and then you take out the tumors, and you see that the tumors that are the control group, they're big tumors, and that's the graph for the big tumor. And the tumors that have been treated with the drug, the mice are treated, but the language we say is that the tumors are treated. So the tumors that are treated with the drug, they're fairly small tumors. They're not gone like the previous one, but they are very small tumors. And so this group shows that the treatment also works for this drug. Treatment will make it small, but it's showing you that if you did prevention would really prevent it. Prevention would make it very, very small. And so that's all I have for explaining to you about um, the tumor models on a preclinical basis. And I will explain more on this when I explain a tomorrow, when I explain on a paper about these in our next topic. So then we will go and look into some tissues, etc. Um, okay, so I want to thank you for this, for being able to describe this to you. And I hope you will now be able to understand that you can even do immunohistochemistry of this one. You can do immunohistochemistry. You can cut these tumors, and you can examine them. Um, and that I will discuss in another section. So, Pooja, Dr. Bhomik, I'll be happy to take questions. So, you also now understand that um, um, preclinical tumor models take 30 days minimum. If this was a rat and we needed to keep it longer, it can go for 6 months. But this is acceptable. The one at 30 days, this is not ideal, but it is acceptable. And so, for lack of better model systems, these are all acceptable and they're all publishable, etc. So I hope
I,
that gave you a perspective. So if you wanted to do immunohistochemistry, you just do it on the tumors here. Thank you, Dr. Bhomik. You wanted to ask something? Sorry, I'm
so I
getting.
think if any kind of question, please ask to madam from the participants. Parvati madam, any question? Okay, and any other from the participants? So if there is no question, we are going to, that is ending this session. And the next session, I think now after 1 hour, we can start. Uh, what is your opinion, madam? In schedule, this is given
Yes.
2 o'clock. So as we can start it in 1:30 after 1 hour.
Yes, please.
Okay. So Pooja, please announce the next session we can start. Is it possible that is prepone the session?
How much you want to prepone? Are you talking to me?
Oh, it is the same link with you have given this link, madam. No issue. Okay. So 2 o'clock. It was the 2 o'clock. The tutorial 8 histopathological imaging. Now it will start from 1:30. Okay. So madam, we will see
1:30,
again.
it means it'll start 1 hour from now, right?
One
Yes, ma'am.
after.
1 hour from now. Now
Okay.
it is 12:30. We will start at 1:30.
Sure.
Okay, so
Thank,
we will proceed in this way. So participant, please active on 1:30.
And if you
Thank
want
you to
to,
all.
if you want to maintain the schedule, you can also do that, you know. I can be here at whichever time if you wanted. Did we finish a little early today, right?
yes, ma'am.
Uh, little early, I
No,
think.
I'm not sure. I'm just saying that if you want to keep
Yes,
the
ma'am.
schedule,
It
you
is
can
a
do.
little early, ma'am. So it's okay, ma'am. We can start at 1:30.
Okay, no issue. From our side, no problem, madam. If you have to go with 2 o'clock, this also no issue.
No, I'm just
No,
saying, if you want to extend your lunch and you have your lunch for 1.5 hours,
Okay, okay. So Pooja, I think
then you can maintain your
we can,
schedule.
yes, yes,
Yeah.
this suggestion also good. So we can start from 2 o'clock as per our schedule. Okay.
Yes. Yeah,
So
I
Pooja,
think that works.
announce it. Announce
oh,
it. Okay.
okay, ma'am. Okay, then we will start our next tutorial 9, tutorial 8 at 2 p.m. So we are ending our lecture 7 session, so lecture 8 session. And thank you, madam.
Thank you.
Thank you, participants.
Okay. All right. We'll join again.